Hi dear students, good morning and welcome back to our Bio-zoology class. We are very happy to inform that you are very cooperative with us and you are following us. Hope you are covering the portions also and kindly follow the same for the forthcoming classes too. Good? Okay. So before we move to the today's topic, let us do one review regarding this previous class. So in the previous class, we have discussed regarding the three objectives, that is three domains of life, five kingdom classification and taxonomic hierarchy. So the first one, three domains of life. As per this, the entire organisms of the earth has classified as three domains, that is archaea, bacteria and eukarya, right? So archaea in the sense extremophiles, the bacteria which live in extreme climatic conditions. Second one is bacteria, and the bacteria in the sense cyanobacteria as well as this bacteria belongs to this bacteria group. And the third one is eukarya, which includes protista, fungi, plantae and animalia. Is it clear? And the second one we have seen it is five kingdom classification system. And as per this system, the entire organisms are classified as five kingdoms. So do you remember? Monera, protista, fungi, plantae and animalia. Is it fine? And the third one we have discussed, it is taxonomic hierarchy. So taxonomic hierarchy means one classification system or classification order is given for each species to identify each organisms or each species is called taxonomic hierarchy. For example, kingdom, phylum, class, order, family, genus, species. As per this level, the hierarchic order will be there. Is it fine? Shall we move to the next class? So we can see today's class. Fine. So today's class we are going to discuss regarding the three matters, that is nomenclature, concept of species and tools of taxonomy. Nomenclature, concept of species and tools of taxonomy. So let us see one by one what is that. Fine. Okay. So below this nomenclature title, I have displayed few figures here, right? Few pictures. Can you name them? Yeah, of course. Peacock, cherry blossom, deer, goldfish, butterfly, tulip flower, right? So now we have called these organisms with one name. So we have identified peacock with its name as peacock, right? And we have differentiated peacock from the goldfish with its name, right? So we have given one particular name for these organisms. That is called nomenclature. Nomenclature is a Latin term actually. It denotes naming. It denotes naming, right? So now we have given the names for these organisms. Now let us see this definition for nomenclature. Fine. So nomenclature definition is the process of assigning scientific names for animals or taxonomic group is called nomenclature. So they say nomenclature is one of the process for assigning scientific names. So we are assigning or we are giving scientific name for one animal or one taxonomic group is called nomenclature, they said. So why should already we have given the name, right? So in the previous slide we have given certain names for the particular organisms. Then why should they have highlighted here the word as a scientific names? So what is the use of this one? What is the need of this one? So why should we have give one scientific name? Some reason will be behind this. Am I right? Okay. So let us see what is this reason behind this. So nomenclature definition is the process of assigning scientific name for animals or taxonomic group is called nomenclature. So let us see what is there behind this scientific name and why should we have to give the scientific name? What are the rules we have to follow the scientific name and how can we assign this scientific name? That details we are going to follow. Clear? First one, types of nomenclature. What are the types of nomenclature is there? The first one, vernacular nomenclature. Second one, binomial nomenclature. And the third one is trinomial nomenclature. So vernacular nomenclature, binomial nomenclature, as well as trinomial nomenclature. And let us see in detail what is this each varieties. Fine. So the first one is vernacular nomenclature, which means the naming of these organisms with regional language or local language is called vernacular nomenclature, right? So here I have displayed one animal, right? And nearby that I have given this name of this animal with 5 languages. The first one, the animal is called as Yanai in Tamil. Then in English it is called as elephant. In Malayalam it is called as Anai. Then in Hindi it is called as Hathi. The same, Telugu it is called as Enugu. Do you notice one thing? The animal is same, but we have given 5 names for the same animal. The organism is same, but we are calling the same organism with different names. And do you know one thing? The report says there is more than 6,000 different languages are there throughout the world. If you take India, 22 official languages are there, and again the report says more than thousands of local languages are there, right? In the sense, the same organisms may get more than 6,000 names. Understand? So, to avoid this confusion, scientists has planned to give one particular name or common name for one particular species, and it should be followed throughout this world. Is it clear? So that's why they started to assign the scientific names. So the scientific name is common throughout this world. So every people can understand the same animal with this name. Is it clear? And this naming will be given as per the guidelines of the organization, that is ICZN, that means International Code for Zoological Nomenclature. As per the guidance from this organization, the naming of these organisms will be done. Is it clear? So vernacular nomenclature means assigning these names with local language. So there is a lot of drawback we can found, and that's why they are assigning the scientific names. So let us see how can we give the scientific name for these organisms. Is it clear? First method is binomial nomenclature. So with this term even you can understand, bi in the sense two and nomen means name. So nomenclature already we said naming of these organisms. So binomial nomenclature means as per this method each and every organisms or species must to have 2 names. Right? So one name is a genus name and the second name is a species name. So genus name must begin with a capital letter and the species name should be with a small letter. So hope these details you know already, right? And this method is introduced by the person Carolus Linnaeus. Even we are following the same method even now also, right? So here 2 examples I have given. Our national bird peacock, its scientific name is Pavo cristatus. So 2 names are there. Is it clear? And our national animal tiger, its scientific name is Panthera tigris. So 2 names are there. Understand? Okay, next one. Trinomial nomenclature. So trinomial nomenclature means the same organism or particular species will be called with 3 names. So tri means 3 and nomen means name, right? And this method is introduced by the person Huxley and Strickland. Clear? So here the same method only followed. Genus name and the species name only will be given. For example, this is the figure I displayed here. This is house crow. Its scientific name is Corvus splendens splendens. So as per the rule, we have to give only generic name and the species name. Here also the same. Corvus is a generic name and the splendens is a species name. So splendent in the sense this particular species may come under, the particular organisms may come under the same species splendens. but among them even little minute differences will be there. That's why they have made it under a subspecies. So that's why subspecies name also will be included. So here the subspecies also splendens. That's why it is getting the name Corvus splendens, which means first name is Corvus is genus, splendens is species, and splendens is again subspecies. So the organism is getting 3 names. Is it clear? That method is called trinomial nomenclature. Fine. And here they are introducing another one new term. It is called tautonymy. Tautonymy, which means the practice of naming the animal in which generic name and the species name are the same is called tautonymy. Do you get me? One example I have given here. This is the scientific name. Naja is the scientific name for cobra, Indian cobra. Here, the first name Naja is a species, genus name, and the second name Naja is a species name. Here, genus name as well as species name both is same. Such a variety is called tautonymy. This kind of naming is called tautonymy. Is it clear? So, it's one of the 3 mark question. So, the practice of naming the animal in which generic name and the species name are the same is called tautonymy. Is it clear? So, 3 method of naming we said: vernacular nomenclature, binomial nomenclature, as well as trinomial nomenclature. And this also one of this term, it is included under this naming method. Is it clear? And we can go to the next topic. Rules of nomenclature. Rules of nomenclature means when we assign the scientific name for one particular species, certain rules we must to follow. So which is actually it is given by ICZN, as we said already, International Code of Zoological Nomenclature. As per their guidelines only we can assign the name for these organisms. Is it clear? Let us see few rules given by these persons, these organizations, right? Let us see the first one. The first rule actually, the scientific name should be italicized. Here one example I have given, Homo sapiens. It's a scientific name for human, right? Do you notice this one? When we give this scientific name as the printed form, so in the system now, in the computer now, we are giving as a printed form, right? When we write this scientific name as a printed form, the font should be italicized font. Their italicized font will be there. So the italicized font only we have to write. We have to follow. That is the first rule. The scientific name, if it is in the printed form, it should be as a italicized font. Have you got me? Okay. Second rule, if it is handwritten, if we are writing this name with our own hand, if it is handwritten, and genus name and the species name should be underlined separately. That I have given the example there. Homo separately we have to underline, sapiens separately we have to underline. Sorry. Next one. And as we said already, as per the binomial nomenclature, the genus name and species name should be there. So organisms must give either 2 or 3 names. So it should be as genus name as well as the species name. Genus name must to begin with a capital letter and the species name must to begin with a small letter. Is it clear? And next rule is the scientific name of any 2 organisms are not similar. So each and every species has a unique name. It is separate name. It is not there for any other organisms in this world. So that's why they said scientific names are not similar. Each and every species will be denoted as different names. Have you got me? Okay. And the next one. When one scientist is giving the name, publishing the name for one organisms, his name also will be included along with after the species name. Right? For example, lion. Lion scientific name is Felis Leo. But this name is first published by the person Carolus Linnaeus. So after this species name, we cannot give the full name as Carolus Linnaeus along with the year. Right? That's why in the shorted form they are writing Felis, Leo, Linn, full stop, comma, as followed by the year 1758. Do you get me? Actually, this name for this lion is first published by Carolus Linnaeus. That's why his name is included after the species name, but in a short form. Have you got me? Example, Felis Leo, Linn, then one full stop, then comma, then the year of publication, that is 1758. Or again with the short form, instead of Linn, we can write L alone. So Felis Leo, capital L, full stop, comma, then the publication year, it is 1758. That also one of the rule, right? And the another one is, if the scientist who published the name at first can give their name as the species name in place of the species name. For example, Cetodactylus is a scientific name for the ground-dwelling lizard. It is a newly found ground-dwelling lizard. Its scientific name is Cetodactylus. This is found by the person Varadgiri. Varadgiri has found this one. That's why the name of this organism, scientific name of this organism is Cetodactylus varadgiri. But one rule has included along with this. If the scientific name is included, at the end, either we have to add one I or double I or AE. Do you get me? Cetodactylus varadgiri we can write, but at the end we have to include either one single I or double I or A and E. Understand? Okay. So these are the few rules we have explained now to give, we have to follow when we are giving the scientific name for these organisms. Do you get me? Few rules. This is one of the very, very important questions we have to learn. Clear? Shall we go to the next topic? The next topic is concept of species. So in the last class also we have said regarding the species. Species is a group of organism morphologically similar, and it will do the reproduction among themselves. It will do the interbreeding among themselves. It will produce a fertile offspring. These are the characters we have learned regarding the species in the last class even, right? Do you remember? Okay. And here this topic says, few scientists has given their own definition for species. Okay, shall we discuss? The first one is given by the person John Ray. In his book, it is Histora Generalis Plantarum, which was published in the year 1693. Right? Okay. Let us see what he has said for about species. He is telling species is a group of morphologically similar organisms arising from a same ancestor or common ancestor. So one species means we can say, for example, human beings are one species. So morphologically we are similar and arising from the common ancestor. Human beings are arising from this. Of course, human beings scientific name Homo sapiens, we said. Homo sapiens are arising from and Homo erectus. Erectus is arising from Homo habilis, right? The ancestor is same. So that is his concept. He was telling species is a group of morphologically similar organisms arising from a common ancestor. Do you get me? Okay, next. Next term is given by the person Carolus Linnaeus. In his book, it is Systema Naturae. He has defined the species as a basic unit of classification. Of course, he is the father of taxonomy, already we know. That's why, so the taxonomy, as per the taxonomical level, he has given the definition also. So when we frame this classification, as per the species level only, the classification will be made, right? So when we give this species name only, we can exactly found one species, right? That's why he is telling species as a basic unit of classification. It's given by Carolus Linnaeus. Is it clear? Okay, the next. The next person is Charles Darwin, and he has published the book called Origin of Species in the year 1859, and he is one of the familiar evolutionists. That's why he is connecting this species word with the evolution. Okay, and that's why he was telling evolutionary connection of species by the process of natural selection. He is connecting the species with the evolution with the help of this, hierarchic tree or evolutionary tree, we can say, right? Evolutionary tree. So where from this is evolved, one particular species is evolved from which species. So he is connecting the species with the evolutionary path. That's why he is giving evolutionary connection of species by the process of natural selection. Is it fine? Okay, the next one. Okay, so till now we have completed two topics. The first one is nomenclature. So in nomenclature we said this naming of these organisms, vernacular naming, binomial naming, as well as this trinomial nomenclature. Then we said the rules of nomenclature, right? And the next topic we said concept of species, various concepts about the species said by various scientists. Okay, shall we move to the next topic? That is tools for study of taxonomy. Taxonomy in the sense, this study of this or classification of these organisms, right, is called taxonomy. So what are the things is helping for this taxonomic study? What are the things is there to help this taxonomical study? That is called tools for study of taxonomy. Shall we go into the topic? The first one, the classical taxonomical tools and molecular taxonomical tools. Third one, automated species identification tools. And the fourth one, neo-taxonomical tools. Next one, ethology of taxonomical tools. And the last one, e-taxonomical resources. Right? These are all going to help for this taxonomy, classify the organisms or to study about these organisms or species. Shall we look into one by one? One is classical taxonomic tools. So, listen me. Taxonomical keys, museum, zoological parks, marine parks, and printed taxonomical tools. These are all belongs to classical taxonomical tools. Classical in the sense we can see we are following from this ancestral period onwards, that we can say, right? Classical taxonomical tools. So taxonomical keys, museum, zoological park, marine park, and printed taxonomical tools belongs to classical taxonomical tools. Let us see one by one what is taxonomical keys and so on, right? Okay. One is taxonomical keys. When we study about one particular organism, the scientist has given some comments or details about the character of these organisms in the form of keys. So if you want to know about one particular organisms, or if you want to study about one particular organisms, if you go through these keys, you can understand about the behavior and character of this particular organism. Understand? One example I can say here. So this what I displayed is one of the method of this taxonomical key. For example, here by this method, the character of this organisms is explained with yes or no type questions. Just one example I could say. For example, we want to know this name of this animal as last one, gorilla. Just we can imagine gorilla. We do not know this animal name. We want to know about this animal, right? In such a case, of course, we will go to the car data area and we are giving this. They will ask the question. We are going to answer as yes or no type. Is it clear? Okay. So we want to know the name for this gorilla now. Is it clear? So they ask the question, we have to answer as per this character of this animal, right? For example, the first question they are asking, the animal which you want to know whether it has a dry skin is the question. So if the question is yes, of course, gorilla has dry skin. So you are telling yes, the answer is yes. Or if the answer is no, they may say it is a salamander. Or if the answer is yes, then you will pass to the next question. And the next question, whether this organism has hair. So you are telling, of course, the gorilla has a hair. So you will answer the question yes. Understand? If you answer no, it may be the lizard. Right? But you are answering as yes. And then you are moving to the third question. They are asking the next question whether it has opposable thumb. Opposable thumb means both the thumbs should be in the straight. That is called it is opposable thumb. So tiger, there is no opposable thumb, you say. That's why if it is no, it may be the tiger. But you are telling it is yes, because it has opposable thumb. And which means they are asking the next question whether it is bipedal. Bipedal means whether two legs are there for movement. So gorilla has 4 legs. So you are telling the answer is no. So your answer may be gorilla. But if it is yes, your answer may be human being. Do you get me? This is the keys. So if you want to study about one known organism or unknown organism, for example, you have one organism, whether you do not know whether this is identified already or the name is given already, these things you do not know. Understand? You wants to know. So they will ask the questions whether it is has this character. You will give whether yes or no. And they will go for the next question whether you have this character for these organisms. As per that, they will ask the question as a yes or no type, and you will answer as per the character of this animal which you have already. Then you could identify the particular animal. This is one of the method for taxonomical keys. Is it clear? So this is helping for this taxonomical study. Okay. Shall we go to the next? Of course, museum, what we can say museum here is a collection of preserved plants and animals. Museum consists of the collection of preserved plants and animals. Even extinct animals and the surviving animals, both will be there in the museum, right? You may visit so many museums, I hope. So their extinct animals also will be there, and living animals also will be there in a preserved condition. So what is the use of this museum? How it will helpful for the taxonomical study? If we wants to study about the character of one particular animal, if we go to the museum, we can closely watch this animal. You can closely observe the character of this animal, morphology of this animal, everything. For example, if you want to know about the one particular snake, you may not go near with that even when it is in a natural environmental conditions. But if it is inside the specimen in a preserved condition, you can go very closely with that and you can watch it. Am I right? So these also helping for this taxonomical study. Is it clear? And the next one is zoological park. Zoological park is different from this zoo, right? Zoological park in the sense the animals are left free in a free environmental condition, but with the care of this human. It's not the forest. Here we can say wild animals are kept free in a protected environmental conditions with the care of this human. So in such a cases, in its own environment, how it is behaving, how its food pattern is there. So these are all the characters naturally we can able to gain. Understand? If it is left free in this free environmental conditions, behavior of this animal, food habit of these animals, all those things, many characters we can able to gain, right? So this also helping for the taxonomical studies. Yeah, of course, like zoological park, here it is a marine park. In the zoological park, wild animals are protected in environmental condition, but here aquatic organisms are protected very well in a controlled condition with the care of this human. Is called marine park. So here also we can observe these animals and its behavior and its movement, its reproduction pattern, everything we can able to observe. And this also helping for this taxonomical studies. And the last one is printed taxonomical tools. So printed taxonomical tools means other than this taxonomical keys or museum or zoological park or marine park, these are all helping to identify particular organisms or to know about these particular organisms. The same, other than these things, the, the entire description regarding one particular species or organisms will be written in a textbooks or books, so which is written by so many others, right? So in their details or description we can able to collect. When we go to the library, we can collect a lot of books, and from there we can collect these details. That is called printed taxonomical tools. Is it clear? So this is the first tool we have discussed now, classical taxonomical tools. So classical taxonomical tools includes Taxonomical keys, museum, zoological park, marine park, as well as printed taxonomical tools. Is it clear? Okay, shall we move to the next? Next one is molecular taxonomical tools. So molecules is maybe the new term you may not be learned in the lower classes. So moreover it is one of the new techniques, current technique also we can say, molecular taxonomical tools. So the previous topic we said museum, zoological park, marine parks, so and books, these are all helping to identify the organisms as well as to know about the character of these organisms. But in the molecular taxonomical tool, the researchers are going in a minute level, even in a molecule level. So exactly we can say DNA level, even in atoms level, the studies are going on. That is called molecular taxonomical tools. Basically, here they are giving the importance to DNAs, right? So do you know the structure of this DNA? Now, generally, we can say DNA consists of double helix and bases will be there and so on, right? So here these are the whatever I have noticed here is recent technologies, current technologies, right? So I will give one outline regarding these things to make you to understand, right? So as per that, the first one is DNA barcoding, second one DNA hybridization, DNA fingerprinting, and RFLP means restriction fragment length polymorphism, and next one PCR, polymerase chain reaction. These are some process or procedure or technologies is helping to identify one particular species. Have you got me? These are the techniques. It's a recent and current technologies. Okay. Just we can give one outlook regarding each varieties. Do you get me? Among these, the first one is DNA barcoding. Have you seen the barcoding? Yeah, of course. When we go to this supermarket, we will purchase these things, and wherever, if you mostly in most of these products, the barcode is given, right? What is the use of this barcode? So the product may consist of one barcode. It may have two different color lines, parallel lines, and right, dark line as well as the light line will be there, and the staff may scan this with this barcode scanner. So when they scan with these codes may consist of this particular numbers will be there. So when they scan, the entire details about this product will be collected by these persons in the system, in the computer, right? That is the use of this barcode. For example, each and every line has one information. Each and every line has one information. For example, manufacturer, product code, cost, worth, etc., etc., right? So each and every line has one information. It will be given as a bar or as a number. So that is the barcoding system. So with this uh, code or this width of this code, this information will be collected about one product. The same method here they are applying for the DNA also, right? So organisms or species may consist of the DNA. This DNA's character is explained as a code. As a code. This code is called barcode. So when we scan this code, barcode, of course about this DNA of one particular species will be learned. Understand? When they scan this code consists of all the information regarding this DNA which is taken from one particular species, either this or any one species. So that is called barcoding system. Do you get me? Okay. Next one. Hybridization. This is also one of the procedure or one of the technology. Hybridization technology means, combination of two variety is called hybrid. Variety 1 is there, A DNA is there, B DNA is there, and it has a double helix. So first procedure it will rewind, so it will separate, each strands will separate, and A strand will combine with the B strand. That's why they denote it as various colors. Can you see? And B strand will combine with the A strand, so a new variety will be produced. That is called DNA hybridization method. So this method also helping for this DNA studies, and DNA studies is helping to know about for the particular species. Do you get me? One is DNA fingerprinting. So fingerprinting, you may know about the fingerprinting. thumbprint we know, right? So we know thumbprint is one of the unique one for each and every individual in this entire universe, entire world, right? Thumbprint is very unique. The same DNA also very unique. So the DNA of one person will not be similar for the next one, right? So that's so like a thumbprint, the DNA printing also one of the unique one for each and every individual other than this identical twins. But the identical twins thumbprint will be different. Do you get me? So that is called, it's a DNA fingerprinting. So DNA fingerprinting means actually if he wants to know about this one, a lot of information is there behind this one. For example, the DNA consists of bases. For example, adenine, guanine, thymine, cytosine. These bases will be there. These bases will be repeated throughout the length of this DNA, right? But these bases, this repetition of these bases will not be similar for individual to individual. Do you get me? Adenine, guanine, thymine, cytosine. These four will be repeated throughout the length of this DNA, but it is not similar for individual to individual. For each and every individual, this repetition will be different. That is called fingerprinting, DNA fingerprinting. So this also exactly helping to identify one particular species without any doubt at all. Have you got me? Okay, the next one. RFLP, restriction fragment length polymorphism. This is also one of this vast procedure, but let me give you some guidelines about this one. Restriction fragment length polymorphism in the sense, so the DNA will be collected, the DNA extract will be collected from one species, then it is allowed for a fragment, which means some restriction endonuclease enzymes will be, some enzymes will be allowed in this sample. So the length of this DNA will be fragmented. The entire DNA will be fragmented as a pieces. Have you got me? With the help of this enzyme, the DNA will be splitted or cutted, okay, with a different length. Then this will be applied to the next one. It is a centrifugation method. So as per the length of this DNA fragment, the sedimentation will be there. It will settle inside this centrifugation tube. So as per this length of this fragment, or as per the sedimentation of this fragment, the character of this segment will be studied. That's what we can say simply about this restriction fragment length polymorphism. So this also helping for this learning about one particular species. Okay, next one. PCR technology, polymerase chain reaction, is also one of the recent technologies. Polymerase chain reaction means the DNA will be multiplied with various numbers, a lot of numbers. Same DNA multiplication will be produced. For example, as we said already, DNA consists of two strands. So these two strands will separate first. Then each strand will produce the matching of another one strand. That is also called as a template strand, that you will come learn in this next classes, right? So two strands will separate each other. Then each strand will produce its own matching strand. So again, one DNA will produce two DNA. Then that DNA will separate again. Then again it will produce its matching strand. So likewise, the process will be repeated again and again. So lot of DNA copies will be produced from the same one particular DNA copy. So this technology they are applying to learn about this particular species. Okay, so these are all these techniques they are applying to study about this taxonomy, that is called molecular taxonomical tools. Have you got me? Shall we go to the next? Now next one is automated species identification tools. Of course, this is one we can say it is a cyber tools, means through internet we can collect these details about one particular organisms. So various websites will be given. So if you go through these websites, we can collect the details or description regarding one particular species. That is called automated species identification tools. So as per that few it is given here. The first one is ALIS, automated leaf copper identification system. If you go through this website, of course we can understand about one character of this leaf copper. Leaf copper is one of the insect which is living on this leaves of this plants, right? So if you go through this website, we can understand this character of this leaf coppers. Okay, and the next one, DAISV, digital automated identification system. Of course, this is also internet form, right? We can collect the details from the net. That's what they said, it's a digital. Next one is ABIS, automated bee identification system. So regarding the bees, we can collect the details from this website. And next one is SPIDA, species identified automatically. So especially spider, wasp and bee character will be analyzed with the help of this data, right? And the next one is drawings. So with this data we can collect the details regarding this bee wings. The wings of these bees will be identified, right? So these are all the databases we can collect from this through the internet. Is called, it is automated species identification tools. Shall we go to the next? The next one is new taxonomical tools. Here some microscopes they are using. So I hope you may know about the various microscope, the dissection microscope, compound microscope, scanning electron microscope. So various microscopes are there. So here I have displayed electron microscope. So even a single cell also will be sliced into various pieces, and each minute details about one single cell even we can understand with the help of this electron microscope. So this electron microscope also helping a lot to know about this each and every organisms. Once ethology of taxonomical tools. Ethology of means as per the behavior of these organisms we can give the classification. That is called ethology of taxonomical tools. For example, as per the sound of this bird also we can classify these birds. And another one example, as per the bioluminescence. As bioluminescence in the sense this organisms will observe this light and it will emit it. That is called, it is bioluminescence. Based on these character also the organisms will be classified. That is called ethology, right? One, of course, e-TAXONOMICAL resources means, of course, it is from the databases we can collect from the internet or computer. That is one of this method. They said is INOTAXA. That means integrated open taxonomical access, which is found in London actually. Then so a lot of informations or data we could able to collect from this resource. One of this example, INOTAXA. Is it clear? Hope you have understood, and just we can go for some assignments. Fine. I give few questions here, dear students. The first question is define nomenclature. Second question, define tautonymy. And third question, define species. So species even with, and three definitions we said. So these are all will comes under this either 2-mark question or 3-mark question. And I will give few 5-mark question also, but it is a very simple question but important questions. For example, one thing, explain rules of nomenclature. It is very simple. Explain classical taxonomical tools, also simple. How molecular taxonomical tools helps in taxonomical study. And last one, explain automated species identification tools. So last 4 may comes as a 5-mark questions, right? So dear students, that you look into the book and read line by line, and these are the highlighted few questions you can cover up for time being, and rest of the things we can revise after the open, and hope you will do well. Until then, we can meet in the next class. Until then, bye. Thank you.